We're going to continue our discussion of cell division by talking about meiosis. Remember that mitosis takes place in all our somatic cells, whereas meiosis takes place in our germ cells, which are the cells that will produce the gametes or the egg and sperm. So, the life cycle of sexually reproducing organisms goes through phases of meiosis and fertilization. So the cycle goes that you have germ cells, you go through meiosis, you make gametes, you reproduce with somebody, and this is going to combine the egg and the sperm to make a zygote. And this zygote is going to be a diploid cell, which means it has two sets of chromosomes, one set from mom and one set from dad. And that zygote is going to grow into a fully fledged organism, and the process will start again with meiosis, going back to the haploid gametes, or gametes that have only one set of chromosomes. So this will take place via meiosis. Unlike mitosis, meiosis has two sets of cell divisions because we have to go from, instead of one chromosome to one sister chromatid, we have to go to a whole pair of chromosomes to one sister chromatid. So we have to divide the total amount of genetic material in 4. So we're going to start with meiosis 1, and this has the same kind of phases as mitosis. It's got prophase, metaphase, anaphase, telophase, cytokinesis. But there's a couple little cool things that happen in meiosis. One of those things is going to be crossing over, and crossing over is a way that we get more genetic variation in sexually reproducing organisms. And this works by, you have a homologous pair, and that's basically the same alleles, like say hair color, eye color, the genes for those are like on a chromosome. You have one chromosome from mom and one from dad with the same alleles, and they're numbered, so maybe like they're both chromosome 1, and they will match up. So like, see how these chromosomes are matching up by their different alleles, so they're aligned, and they are going to undergo crossing over, which means that a little bit of one chromosome is going to trade places with a little bit of another chromosome that is its homologue. And so you get different chromosomes than the ones you started out with from your parents. So this is going to go through prophase. The nuclear envelope is going to break around these chromosomes, and they're going to start attaching to the microtubules. It's going to go through metaphase. So instead of chromosomes all lining up individually on the metaphase plate like we do in mitosis, they're going to line up in their homologous pairs. And then in anaphase, instead of separating out into sister chromatids, these homologous pairs are going to separate into chromosomes. So it's different because you have both your sister chromatids together here and there. And you can see how they're crossed over a little bit. Like there's a little bit of red in the blue and a little bit of blue in the red. And that's anaphase. They're going to go toward their separate poles. And that's going to happen, like we said, via the depolymerization of microtubules. By the way, if you haven't watched the mitosis video, that's kind of a prerequisite to this one because I go through this stuff kind of fast. And in the mitosis video, I go into a lot more detail. Anyways, telophase is going to happen. The nuclear envelopes are going to reform. And you're going to start to see a cleavage furrow, which is those little vesicles that start to separate the cells. And then we will have cytokinesis, cytocell kinesis moving. They are going to move apart. And you're going to have 2 haploid daughter cells. And so you might be thinking, okay, well, they're haploid, so that's exactly what we want. But it's not, because remember, we still have chromosomes with 2 sister chromatids. So the DNA is still duplicated. The only reason that it's called haploid is because you don't have those homologous pairs. But within each chromosome, the DNA is still duplicated, so we still need to go through another cycle of meiosis. And that's exactly what we're going to do. So this diagram shows 2 daughter cells forming into 4, granddaughter cells? I don't know what you call them. But anyway, you have prophase, nuclear envelope breaks, microtubules start to form out of the centrosomes. You have metaphase. The chromosomes individually line up on the metaphase plate. In anaphase, sister chromatids are going to separate, and the microtubules are going to again depolymerize. The tubulin subunits are going to come off at the kinetochore end. And Then we're going to have telophase. Nuclear envelopes start to reform. And remember that in this kind of thing, in meiosis 2 and in mitosis, when we're separating the chromosomes into sister chromatids, as soon as we make them into separate daughter cells, those chromatids are again termed chromosomes. They're only termed sister chromatids for as long as cell division is taking place, just so that we have something different to call them. But anyway, telophase is going to happen, nuclear envelope is going to start to reform, you're going to start to see the cleavage furrow, and then cytokinesis will completely split the cells, and you will have 4 gametes that are either egg cells or sperm cells. The egg cells and sperm cells are going to look very different. Sperm cells are very, very stripped down and basic. They do not have organelles. They have like something called an acrosome, which is like what interfaces with the egg, and they have their DNA, and they have a flagellum, but they don't have anything fancy. All your organelles, your lysosome, your endoplasmic reticulum, your Golgi, your mitochondrion, you get those all from the egg. But they're both going to be haploid gametes, and that's the story of meiosis. I hope this helped. Oh wait, there's a couple more things that I want to go over. So we talked about crossing over as a means of genetic variation, but I also want to point out the law of independent assortment, which is something that you'll probably learn about more if you have learned about Mendelian genetics. So it basically means that traits that are on different chromosomes are completely separate from each other in the probability that each gamete is going to get them. So this has to do with meiosis 1. So do you see how it's not like all the blues line up on one side and all the reds line up on one side? It's that they line up kind of randomly. So the blues could be on the bottom or the reds could be on the bottom, and they're not all going to be the same. Like, here's a red, here's a blue, here's a blue, here's a red, right? So although they line up in homologous pairs, they line up randomly, and that's going to contribute to genetic variation. And then there's, of course, going to be variation in determining which gamete fuses with which gamete to form a zygote, because there are multiple sperms and multiple egg cells depending on the cycle. So that's how we get a huge variation in sexual reproduction as compared to asexual reproduction.